Right, my name is Hammond, and I'm presenting on bactericidal effect of Manuka honey against antibiotic-resistant nosocomial pathogens. Why this topic? Because it's very important that we get into the natural honey, because antibiotic resistance is increasing, and the side effect of antibiotics is also being a problem so far. But then, how can we use a natural product which have less side effects for treatment? So we decided to use Manuka honey. Manuka honey is a honey derived from a plant from New Zealand. So this honey has antibacterial properties, which is made from the honeybee. So then we try to find out whether this honey, we have antibiotic resistance. We expose them to this honey with different concentrations. So we check whether the antibacterial properties in the honey is able to kill the organism, or whether it is able to stop it or inhibit it from growth. So we exposed it with different concentrations, and we got to know that it's able to kill the organism. But then we have different types. We have the medical grade honey, and then we need a table honey, which means the edible honey. So medical grade honey is for topical use only, which cannot be used orally. But then we have an organism like C. difficile, which treatment is oral. So with the oral treatment, we have to find a honey which can be edible. So our test is to use edible honey, not the topical honey. So we try to find out, and we got to know that there is another honey, which is also Manuka honey, which is able to inhibit it. And the difference between the Manuka honey, the edible honey, and then the topical use honey, the difference is not much. Both of them are able to inhibit the organism. We also check, this is the zone of inhibition. So as we change the honey concentrations, we got to know that the zone of inhibition increases. You see the build-up wall, that the organism is not able to cross this border, which means the antibacterial properties in the honey was able to maintain and able to resist the organism. We also check whether is it the sugar content that kills the organism, or is it something different. So we prepared an artificial honey, which contains the 4 major sugars present in natural honey. And we got to know that even at the 50% level, the natural honey couldn't kill it. It just stopped it, but it couldn't kill it. As compared to the 8% of 8 and then 12% of the medical grade honey, and then the artificial honey. No, not the artificial, the edible honey. So at the end, we got to know that fine, it is good. So C. difficile treatment. Now our next, this is the in vitro test. So we are yet to translate it to the natural, to human level, or to the animal, so that it can be productive for everybody to be used. So now at the moment, this is so far where we have got to, but then we need collaboration so that we can do more work to ensure that this will become a reality for everybody to get use of. So we've made some publication has come out, but then we are yet to submit this work for publication. Thank you very much.